Okay, thank you for inviting us to this seminar series. And so me, Anders, and Maria will present the toolboxes that we have in SBDI for handling metabarcoding data. And to give you like a bit of a background, I will start to introduce what SBDI is and what GBIF is. Maybe these are kind of unknown organizations to you. And then I will briefly describe how we handle genetic data in SBDI. We will follow with how we publish, how kind of researchers should publish their metabarcoding data in ENA, and Maria will tell us about the guide that she's developed for this. Then I will briefly describe the NF-core AmpliSec pipeline for metabarcoding data processing that we're using in SBDI. And then finally, Maria will tell us about how you as a researcher publish and do searches and download metabarcoding data from SBDI. Okay, so a bit of background about SBDI, the Swedish Biodiversity Data Infrastructure. So this is a national research infrastructure supported by the Swedish Research Council and co-financed by the participating organizations, which is 11 universities and a couple of government agencies. And the host is the Swedish Museum of Natural History. And SBDI is also the Swedish node of GBIF. And I will tell you in a moment what GBIF is. And what are we doing here? Yeah, we are not generating new data, but we're rather making sure that biodiversity data is being mobilized, and we provide tools for analyzing this type of data. And what type of data are we mobilizing? Well, it's a mixture. It's like citizen science data from bird watchers, amateur botanists, etc. It's data from systematic monitoring programs, both terrestrial and aquatic, from sponsored by governmental organizations. It's data from Natural History Museum. It's kind of general research data from smaller or larger research projects. And this includes genetic data from DNA sequencing of environmental samples, which I will talk more about today. And the core, let's see if I can change slide there. The core or the heart of the SBDI is probably the BioAtlas. And the BioAtlas is a platform where all of these species occurrence data from the various sources are gathered, and where you can then search for a species and get it displayed on a map, and you can download its occurrences over space and time. And this platform is built on the Atlas of Living Australia platform, which was launched already in 2010. Now this type of platform has been, it's now called the Living Atlases. It's deployed in 19 countries, serving as national hubs for biodiversity. It is built on open source software, and it's kind of developed in collaboration between participants in different countries. It is done, and it works then for storage, mobilization, analysis, and visualization of biodiversity data. It has this kind of graphical user interface, but it also has web services, APIs, and R tools that you can interface with the database with. And the nice thing is that these national kind of hubs of databases, they are not isolated, but they're joined into something called the GBIF. GBIF is the Global Biodiversity Information Facility, and it is then sponsored by many governments around the world. And it's kind of similar to then the SBDI and the BioAtlas, that it gathers all types of biodiversity data, but it kind of harvests data from these national hubs. So it's kind of, all gathered inside a GBIF server and platform. But the national hubs, they are a little bit more tailored towards the needs in the different countries. So it's a nice kind of combination of global and national efforts here. And the GBIF has an amazing number of data sets, more than 100,000, and more than 3 billion species occurrence records. And data from 63 participating countries, etc., etc. And if you look at the number of peer-reviewed publications using GBIF data per year, it looks like this. So around June this year, 1,000 publications were using data, and it's estimated to be more than 2,000 publications only into 2024. So the use of this is massive. And here are just 4 examples of studies using this type of data out of the 11,000 in total peer-reviewed articles. So it's mainly studies about then species distributions, how it changes over time, often in relation to climate change. Okay, and the Swedish contribution so far is 160 published data sets and about 135 million species occurrences. However, this number is expected to increase dramatically during the coming year, since we will put in a lot more metabarcoding data, and for these data sets we have many species occurrences. Yeah, okay, so that was a bit of a general background about SBDI and GBIF. And now we'll more focus on the kind of metabarcoding part, which we in this group are responsible for contributing with in SBDI. So I guess all of you, or most of you, know what metabarcoding is, but if there's someone that doesn't, I will very briefly just tell you how it works. So basically, you start with an environmental sample, you extract the DNA, all the DNA from this sample, you run a PCR using some using primers that target the group of organisms, say all the bacteria or all insects in your sample. Then you sequence these PCR amplicons. When I started with metabarcoding in 2005, we were happy to have, say, 1,000 sequences per sample. Today, we often aim for 1 million sequences per sample because the sequencing costs have dropped so dramatically. Then when we have all this data, we remove errors in the sequencing, and we retrieve these amplicon sequence variants, which then should be kind of true biological sequences that can differ as little as one base between them. Then when we have this kind of list of ASVs, we do a taxonomic annotation of them using a suitable reference database. And then we get this, often we do this on many samples, so we get counts of the different ASVs in the different samples together with a taxonomic annotation of the ASVs. Then, of course, you can also aggregate this data at different taxonomic levels. So you can sum up all ASVs belonging to genus, for example. Okay, and how does the data flow in SBDI look? So basically, you start with getting your sequences, often from a sequencing provider like NGI. They deliver the sequences to the researcher. We are not actually storing any raw data in SBDI. There are excellent repositories for this. It is, though, a bit tricky to submit the data, and especially the metadata. And for this, Maria has developed a guide that she will tell about soon. So we expect the researchers to submit their raw data to ENA or NCBI. And once that has been done, the sequences are being denoised, so you can reconstruct these ASVs. And for this, one can use a tool of choice. And we recommend using the NF4 AmpliSec pipeline that we have been co-developing. And I will tell a bit more about that soon. And then once you have your ASVs, you are ready to submit them to us. And for this, so you submit them to what we call the ASV portal, and Maria will tell you about this later. And so you basically submit your ASVs, their counts, and the contextual data. And they get integrated in what we call the Swedish ASV database. But they are also then re-annotated, so we get the uniform annotation of all the data that are in SBDI, and they are then also integrated in the BioAtlas. And if you opt for, they are also submitted to GBIF. And then we have developed tools for then searching for data in the ASV database. You can use a blast search, you can do a taxonomic filter search, or you can search for a specific primer set, etc. And then you can retrieve the corresponding sequences and their occurrences in the bioatlas. So you kind of see where they are found on the map. We have also developed this new R package where you can very easily download data directly from the ASV portal and import it into R and directly do downstream analysis of the data. And we will demonstrate how to do that as well. So I think that, okay. And why then would you publish metabarcoding data in SBDI? Well, first of all, your data can be reused by others, which is a great thing. So you basically comply with the FAIR principles. Your data can be easily compared with other metabarcoding data sets and also with totally other types of data in the bioatlas, which is kind of unique. Your submitted ASV will be regularly re-annotated by SBDI as reference databases are updated. So, I mean, you submit your data, and then a couple of years later, you can retrieve them back and having a fresh annotation of them. SBDI is integrated in GBIF, so your data will contribute to global knowledge on the biodiversity, which is really cool, I think. And then your submitted data will get a DOI that can be provided in publications. So it will kind of complement the accession number that you get for the raw data in a NCBI. Here you get the DOI that you can use to kind of track the ASVs. Okay.
Okay. So just to reiterate what Anders said, you should notice that SBDI will not store your sequence reads, but we do provide a guide on how to archive them with the ENA. And I'll just start by showing you how to find this guide. And the easiest way is probably to search for the Swedish ASV portal. And the ASV portal is our web interface for metabarcoding data in SBDI. And I'll tell you more about it later on, but for now, just notice that there is a submit option down here, which will take you to an overview of how to submit data to SBDI, where the first step is to submit your raw reads to ENA using our guide. So if you click on the link here, you will be presented with our guide. And this is largely a summary of ENA's own extensive documentation, but we thought it would still be useful with some step-by-step instructions tailored specifically for metabarcoding data. We don't have time to go through it in detail today, so I will just highlight a few things. First, notice that there are 3 different routes for submitting data to the ENA, and we describe the interactive submission, which is done in the so-called web-in portal. So if you head over to the ENA and log in to the web-in portal, you will see a graphic of their metadata model, which gives a pretty good summary of the submission process, where you first register a study, then add samples to that study, and finally associate sequencing experiments and read runs and read files with those samples. We describe all of these steps in the guide, of course, with some emphasis on how to document your metabarcoding samples in a useful way. So, for instance, we recommend which so-called checklist you should use to describe your samples. And then we give you some pointers on how to interpret checklist fields in a metabarcoding context. So, for instance, we explain how to set taxon ID and scientific name for an eDNA sample, and also what ontology-linked attributes are and where you can find these, and many more concepts that may be confusing for a first-time data submitter. Right, so now you know how to find our guide for archiving your raw data with the ENA. So Anders will now tell you how you can proceed and denoise those raw data into ASVs.
Yeah, so for this, we recommend then that you use the NF Core AmpliSec pipeline, but you can use a pipeline of choice. So the NF Core AmpliSec pipeline is co-developed by SBDI, and it has a lot of functionality for processing Amplicon data. So it both for pre-processing, for inferring ASVs, for taxonomic classification, a number of different tools for that, as well as a number of different reference databases. And then you get outputs in a lot of different formats, both standard kind of tables, etc., but also you get the visualizations of the data. And so what's the good things with this pipeline? Well, it's super easy to install. It's platform independent. It's very easy to run. And it's continuously developed through the NF Core collaboration. And it's also very easy to do research reproducibly because you get these files that have kind of annotated exactly what you have done. And how do you run it? Well, there are some different options for how to do this. And the prerequisites that you need to have is to have Nextflow installed on your computer, and then you have to have either Singularity or Docker or even Conda installed. And if you have that, you're fine. But if you want to run it offline, you also need to first download the workflow. What do you have to do to prepare the data? Well, you locate your raw data, and you should ideally make a sample sheet, and then you have to know your primer sequences, and then you define what parameters to use. And you can also collect, this is an optional step, the metadata that you want to submit to SBDI later. If you have that in a file, it will automatically produce output format that you can then use to submit to SBDI. So this sample sheet looks like this. You have the sample ID, and you have the location of the forward reads, the location of the reverse reads, and then you just specify what run they were running if you have done multiple sequencing runs. Since you put these parameters in a YAML or JSON file, you get a very reproducible way of doing your analysis, and it's very easy to run again with the same settings. So how can a command look? Well, this is an example. Nextflow run NF Core AmpliSec, and then you specify the version, unless you just want to use the most recent version. And then you specify a profile, and then you specify this params YAML file. And this last file looks something like this. You have the sample sheet, and you have the forward and reverse primers, and you have, if you want, you can specify where I have put the metadata in this TSV file, and then you specify the out directory. And whether if you want to make an SBDI export or not. Well, if you want to run on Dardel, there is this new profile, PDC_KTH, which makes the run smooth on Dardel. So there are a number of these predefined profiles that you can use for running on different compute infrastructures. But if you really don't like to work on the command line, you can also use this graphical user interface and actually set these different parameters using this manually this way. And once you've done this, the software will automatically produce this YAML file for you. And then it's very easy then to run it. Okay, and how does the output look? So this is an example if you opt for doing the SBDI export option. So you get the different ASVs in the different rows here. You have their taxonomic annotations, the sequences, and you have the counts of them in the different samples. And the metabarcoding data types that we are currently supporting in SBDI, or actually data sets that are included at this point, are for archaea and bacteria, where we are using the 16S RNA gene as a marker and the GTDB database. We have data for fungi using ITS sequences and using the Unite database. For eukaryotes, we have 18S RNA sequencing data using PR2 as reference database. And for more specifically then for metazoa eukaryotes, we use CO1, the marker and the BOLD database. But other groups can definitely be included upon request from users.
Right. So assuming now that you have denoised your data into ASVs, you can then go ahead and submit these to SBDI together with any contextual metadata that you may have collected. And I will show you how this is done. So first, let's just go back to the ASV portal. And again, we can click on Submit. And we have now finished the first two steps. That is, we have submitted the raw data to ENA, and we have denoised them into ASVs. And this means that we can proceed to step number 3, which is to download a data input template. So if we download and open up this template, it looks like this. So in essence, our template is a spreadsheet with tabs that corresponds to a slightly modified so-called Darwin Core archive. I won't go into any details here, but the Darwin Core archive is a standardized format for exchanging biodiversity data. And we have just modified it a bit to make it easier to use for metabarcoding data. So in this context, an event corresponds to a sample, and here you basically add info on where, when, how, and by whom samples were collected. Then there is a DNA tab where you describe your sequencing experiment and processing. So among other things, we want to know which primers you used and how you denoised your data. You also have the option to add contextual data into one of the so-called extended measurement or fact, or EMOF for short, tabs. So for instance, you may have measured salinity and temperature of a seawater sample, and then you would add those data here. And finally, you add your ASV sequences, taxonomy, and recounts into the ASV table tab. Notice also that there is a guide tab where we explain all of these fields and give some example values for these. Right, so when you've filled in your data, you will go back to the submit page and then click on upload to finish your submission. And after that, we will be signing a publishing agreement with you, and then we will publish your data. And this means that your ASVs will be available in the ASV database, of course, in this portal. But we will also produce the so-called Darwin Core archive that includes the occurrence data, which is then pushed to the Swedish Living Atlas instance, which is called the Bioatlas, and optionally also to the international GBIF network. And this means that a dataset published in the SBDI Bioatlas, like this one, can also be made available in the GBIF platform, which includes data from the whole world. Okay, now that data have been properly published, I will go on to show you how users can search for data in our ASV portal. Again, if we go back to the start page, you can see that there are two search options listed as BLAST and filter search. And we will have a look at the BLAST option first. So this is the BLAST form where you can paste in your own sequences here. that you want to align with sequences in our ASV database. You can also set two basic parameters for the BLAST algorithm, which are the minimum identity and the minimum query coverage. And then if you click BLAST, you will see a result table where you can click and show individual sequences. And you can also download the BLAST result table directly in different formats. Or you can show associated occurrence records in the BioAtlas. But right now, I'm going to skip to our second option for searching instead. So we select the filter option here. And this will take you to the filter search form, where available search filters are grouped into sequencing and taxonomy related items. And after combining some values in a few of these filter boxes, you can then click on filter. And we now see a similar result table. And this time I have selected some of the matching ASVs here. And I can then go on to show occurrences of these ASVs in the BioAtlas by clicking here. So this takes us to the BioAtlas platform, where we can map occurrences of these selected ASVs like this, or we can list individual records by clicking there. So the record list looks like this, and we could, for instance, select this specific occurrence of recognized archaea genus to see all the metadata relating to that occurrence. So for instance, here we can see a link to the raw data in ENA. And we can also see how SBDI has re-annotated this sequence, as Anders said, using the AmpliSec pipeline together with a certain version of the GTDB reference database. And as I think Anders mentioned, this is something we do as we ingest your data to ensure that a single ASV only has a single taxonomic annotation in our system at any point in time. But we also update this annotation when new versions of the reference database is released. But notice that your original annotation will also be available here. Right, so now we've seen how data and metadata are displayed in the BioAtlas, but what if you want to do some analysis on the data? Well, for now, we recommend that you download data directly from the ASV portal and then use our small R package to unpack, merge, and process data into a format suitable for downstream analysis. So first, head back to the ASV portal start page, and now we can click on download. And this will take you to a page where you can download complete datasets as condensed Darwin Core archives. So first we can search for 16S data here, and then select the datasets we want. And these are two datasets from the Baltic Sea that we may want to compare. So we can click on download, and then you can also click on the link up here to get some info on how to install our R package. So our R package is called ASV occur, and it includes functions for unpacking and processing datasets that have been downloaded from the ASV portal, and can also be used for aggregating read counts at different taxonomic levels. Note that you install it directly from our GitHub repository by following a few simple steps outlined here. Okay, so if you use RStudio, for instance, it will look something like this. After installing ASV occur, you can list the available functions, And you can also see some help on a specific function, such as load data. And if you then load data from a specified path, you can see that these are unpacked into data tables arranged in a list of lists over here. And if you take a closer look at the ASV list here, you can see our two data sets that we selected, where one has just over 40,000 ASVs and the other has around 3,000 ASVs. So next you can go on and merge loaded datasets. And if you have a look at the help here, you can see that you can either select an array of datasets to merge or just merge all loaded datasets. The resulting merged object will then contain a single table for ASVs. And judging by the row count here, we can see that it is less than the sum of rows in the separate datasets. And in fact, the two original datasets shared around 2,000 ASVs. And at the same time, events and contextual data and so on have also been merged into single tables. Now, assuming that you're interested in the broader picture here, rather than the abundance of individual ASVs, we also include a function for summing read counts by clade within taxonomic ranks. And you can do this both using raw counts and by using values that have been normalized per sample. And as you can see here, we then get a new table for each taxonomic rank. So for instance, here we have one that aggregates data at family level. Right, before we have a look at one of these, I should just say that ASV occur is based on data table functions, which are really efficient for large data sets, but we also include a function for transforming these results into data frames, if that is what you prefer to work with in R. So here I have applied this conversion function, and here is one of the resulting data frames, which shows normalized read counts per family for each sample in our merged dataset. So these are the functions that are included in our R package. And now Anders will wrap this up by showing you how to make a nice visualization of these data.
So starting with the same dataset, having it loaded the same way as Maria did, I will do a couple of kind of downstream analysis, how we can analyze the data. So just one slide about the background about these studies that we have loaded. So both are from the Baltic Sea. One is the smaller one with 21 surface water samples from 2013. The other one has 341 surface water samples, and these are from 2019 and 2020. Now let's see what we can get out of the data. First, we can have a look at this events object where the most basic information about the samples are stored. And these are the same for all studies have this type of data that are in this object. So the event ID, it's kind of the sample IDs. It has both the study name first and then the kind of sample name after the colon. And having this information, we can make a vector of the indexes of the two studies. So one vector for the 2013 study and one for the 2019 study. And that will be useful soon when we're going to plot the data. We can also extract some more data from here, the event date. And using that, we can build this year day vector and a month vector. And we can also find latitude and longitude information. Well, papers, they talked about the salinity gradient, and salinity doesn't seem to be in this object. But then we instead look at this extended measurement of facts object, which Maria mentioned before. So here the users have provided kind of study-specific metadata. And let's see now, salinity is present both up there. And if we read a bit in the methods in these papers, we see that one of the studies just used salinity and another one used the average salinity. So we make this salinity vector where we gather this information. And now we have then, once we now have this basic metadata information, we can first of all plot the samples on the map. So now we plot the samples from the 2019 study on the map, and we color them by day of the year. And then we plot the data for the 2013 study, which was a much smaller study, just including data from 1 month. Okay, and now that we've done that, we may want to do some type of ecological analysis. And what I've done here is just a PCOA on the Bray-Curtis distances. So often in ecology, you calculate this pairwise community dissimilarity matrix using the Bray-Curtis index. And we do that first. And after that, we can do this PCOA, showing how the samples are kind of related to each other in terms of community composition. And now I'm plotting only the 2019 data, and we see that the samples are very much influenced according to salinity, because you have the higher salinity samples to the right and the lower salinity to the left. But you also see that there is a strong seasonal signal in the data. And PC1 is the component that describes most of the variation here. So salinity seems to be the main driver. And now if we add now the samples from the 2013 study, we get them located. So those are the squares. They seem to be located about in the same way. So salinity again seems to be the major driver, and there is also a seasonal signal. Maybe they're a bit offset compared to the 2019 study, which may be because of true kind of biological variation between the years, or it could be due to some technical artifacts in how the samples were collected, etc. So this kind of shows you how you can, with this R package, very easily compare different data sets. Now you might wonder, okay, these data sets, we had quite a bit of overlap in the ASVs, and this is because exactly the same PCR primers were used for generating this data set. What if we use different PCR primers, so you cannot really compare the ASVs? Well, then I would recommend that you maybe do the analysis at the genus level instead. And actually, on this slide, I have calculated Bray-Curtis distances at genus level and plotted those against the Bray-Curtis distance at ASV level. And overall, it's quite a strong correlation. So basically, you could do this analysis at the genus level instead, which should work pretty well then if you use different primers for the studies that you compare. Okay, so that was the last slide about this ASV occur. And maybe I will just round off by giving you some future plans that we have in SBDI for genetic data. Well, we are happy to support more marker genes when such requests are coming in from users. We are planning to integrate shotgun metagenomics data in SBDI Bioatlas using the ENA Magnify pipeline to kind of recruit the data from ENA and convert those into taxonomic counts. We have started that process. And then final, more long-term plan is to make an integrated data flow from NGI to SBDI so that users do not have to do so much kind of manual work. So this is a long-term plan. And by that, we would like to thank you very much for listening, and you're now most welcome to ask questions.